Hi guys, welcome back to another video of Tamba's Guide to Not Effing Up A-Level Biology. Today is going to be a pretty simple video. It's going to be on the structure of the neuron. So this is going to be part of the homeostasis, electrical impulses kind of topic. There's a lot of mechanisms in this topic, but the thing is, they're all pretty overlappy. A lot of the stuff that you learn is going to apply to other parts like muscles and synapses and stuff. So it's pretty interesting stuff. So let's get started. There's two types of systems in your body. The first part is the hormonal system, and you have the neuronal. And they're pretty self-explanatory. They should be like GCC revision. So hormonal is based off chemical signals that travel in the blood, whereas with neuronal communication, it can be chemical signals, but mainly it's electrical impulses. You will see that in later videos that the electrical impulses are due to chemical signals doing their magic, but for this all you need to know is electrical impulses travelled by neurons. Another thing you need to know is the chemical signals act on specific target cells. So, like insulin, for example, it would act on, I don't know, some kind of cell in the body that will cause it to carry out that change. So, act on specific target cells. And with this, with neuronal, it again is on specific cells, but with regards to hormonal, they're usually on a more bigger scale. So, for example, changes can occur across the whole body. And they also tend to be long-term and also long-lasting. If you think about puberty, for example, around the ages of 10 or 11, the body starts releasing testosterone. And that's a hormone that effectively regulates the growth of pubic hair or the development of sexual organs. So as you can see, those are long-term, long-lasting effects of testosterone, which links into the hormonal system. With neuronal, however, it's usually short-lived, and it's also very precise, as in very localized, that's the word. Responses. So let's compare them again. So hormonal, chemical signals that travel in the blood, act on specific target cells. Changes can occur across the whole body, and they're long-term and long-lasting. Often, put that often there, because you'll see again in this topic it's not always a long-term thing. Neuronal, electrical impulses traveled via a neuron, short-lived, very localized responses. Electrical impulses, you'll see them being called action potentials, and we'll see why, because this is a very important term, this topic. Excuse the ugly box. So yeah, these are the two types of I like to think of them as delivery systems in the body. I know a lot of people are going to call me out, especially the teachers, but that's the way I like to think of it, and that's how I'm going to teach myself this topic. Anyway, so the next plausible area we should start looking at is the structure of a neuron. And I know this is going to be a lot of GCC revision, but there are some A-level specific things that you need to know. And it's pretty key if you want to pass the subject. Ooh, that was ugly. So I'm going to draw in a nucleus here. I'm going to draw a few branches over here. And a few more over here, the little dots. Now this part in the middle should be pretty familiar. That's called your axon. And you can think of the axon as basically the region in which electrical impulses are propagated. There we go. Over here is your nucleus, obviously. Don't know that you're screwed, but over here we have your dendrites, and the small parts over here, these are the ones that are actually called dendrites. The big thing over here is called a dendron. And these are effectively small fibers that branch off and carry impulses towards the cell body, which leads us nicely on to the cell body itself. Cell body. Pretty self-explanatory name, I mean, cell body, it's just like the main part of the cell, really. So yeah, cell body's over there. What else can we label from this diagram? These are your axon terminals. And with terminal, you can always think of it as the end of something. So in this case, it's the end of the neuron. I think the way the textbook defines it is where electrical impulses travel away from the cell body. The textbook often defines the function of certain things with respect to the direction in which they're moving. So, for example, with dendrites, electrical impulses are moving towards the cell body. In this scenario, where we are talking about the axon terminal, electrical impulses are moving away from the cell body. And that's going to effectively pair up with other neuronal cells. One more thing you'll know that I've missed out is the myelin sheath. And this is because we have to look in at a bit more detail when looking at the myelin sheath. So what I'm going to do is I'm going to draw out a big myelin sheath and we're going to start labeling it. So let me do it like this. So here we have the axon. And here we have a few weird looking blob kind of things. This is pretty different from GCSE where you're like, oh, myelin sheath insulates the axon and makes impulses faster. It's a bit more complicated than that. So first of all, we have, let's label the axon just to make it pretty self-evident. That's from cell body, and that's towards axon terminal. These little blob things, we call them nodes of Ranvier. So, nodes of Ranvier. And they're made up of a special lipid, which, as you've guessed, is myelin. So, the actual square blob thingy here, the whole thing isn't myelin. It's actually the membrane of a cell called a Schwann cell. So I'm just going to draw a few really ugly, that looks evil. Like a bit of a face. I like that. Yeah, let's keep it. So this is a Schwann cell, named after a scientist called Schwann. Yeah. Schwann cells, and their membranes are rich in myelin. So I'm going to move over here. And remember, myelin is a lipid. And what it effectively does is your GCSE kind of thing, insulates nerve impulses so they travel faster. Also, I just realized a crucial mistake in my labeling. that is not a node of Ranvier, it's actually the gap. Apologies. So the gap over here is your node of Ranvier. Sorry about that, but you know, we all make mistakes. It's 11 at night and I'm really tired. So these are your node of Ranviers, and we define the node of Ranvier as an unmyelinated axon region. And we're going to learn later in this topic that because this region is specifically unmyelinated, it can lead to something called saltatory conduction. I'm not going to delve into it too much, but all you need to know is that the node of Ranvier is basically an unmyelinated region of the axon itself. So this is a quick depiction of what it actually looks like. I'm just going to recap quickly. You have your axon, you have the Schwann cells, which membranes are rich in myelin, and the myelin helps nerve impulses go a lot quicker. The region that is unmyelinated is called node of Ranvier, and you have multiple of them, so it's not just one node of Ranvier. It happens all throughout the whole axon length. And that's pretty much it, actually. I told you this is going to be a quick one. That's pretty much it. The next video is going to be on action potential, so see you then.